Sir, good morning. Hello.
Good morning, sir.
Good
Good
morning,
morning.
sir.
Good morning. आवाज येतोय क्लिअर? हॅलो.
Yes, sir.
Yes, sir.
Yes, sir.
Yes, sir.
So we'll start today. Slide दिसते ना?
Yes, sir.
Yes.
Yes, sir.
Yes, sir.
So we already seen the many methods to see the what is the efficacy of the disinfectant. Means how much concentration we can use as a disinfectant to inhibit the or kill the microorganisms. So this is the main concepts of that, and we have seen the many methods can be used for that to determine that how much concentration or disinfectant is required for killing the microorganisms and to what time. So how much time required for kill the microorganisms? So both this concentration and the time will be determined with the help of this disinfectant concentration. Understand it? So this disinfectant, whatever the test solutions are there, we can comparing with the phenol as a standard solution, right? And comparing the result with this phenol. We have seen the radial Walker test again. So if the radial Walker test, hello. हॅलो कोण बोलतंय मध्ये? हॅलो, आवाज येतोय मध्ये कोणाच तरी? हॅलो, आवाज येतोय?
Yes, sir.
येतोय ना? मध्ये मध्ये कोण बोलतंय? घरातले काय? Okay. So radial Walker test. The radial Walker coefficient is 1. Means that is the same efficacy with the phenol. जे काही तुमचं टेस्ट सोल्युशन असेल, टेस्ट डिsinfectant असेल, if you are comparing with the phenol, and if the radial Walker coefficient is 1, right? So it means that the effectiveness is the same as to the phenol, right? If the radial Walker coefficient is less than 1, so it means that your test substances which are less effective than the phenol, as like understand it. So this is called as a radial Walker test. Then, so today there is a minimum inhibitory concentration. So in short, it is called as a MIC, right? So MIC. So meaning is that, right? So with the lowest concentration, which gives or which kill the microorganisms. So you can determine that how much concentration is required for to kill the organisms. Understand it? So which is the lowest concentration. So minimum inhibitory concentration with that minimum concentration to inhibit the microorganisms. So that concentration should be determined. Understand it? So because what will happen if I use the minimum concentration must be, it should be know that minimum concentration required for inhibit concentration. राईट. समजा तुम्हाला माहित पडलं की ह्या 10 ml ला कॉन्सन्ट्रेशनला 10% कॉन्सन्ट्रेशनला इट कॅन किल द मायक्रोऑर्गॅनिझम्स, राईट? अंडरस्टँड इट? सो बट इन्स्टेड ऑफ द 20% यूज करायची जरूर नाहीये, बरोबर? मिनिमम 10% ला इनबिट होतात, तर 20% कॉन्सन्ट्रेशन यूज करायची जरूर नाहीये. अंडरस्टँड इट? So you can determine that how much minimum concentration which gives that effect. Understand it? So this is called as a minimum inhibitory concentration. So this is a disadvantage of that test is the number of organisms will be considered too low. Depending upon the concentration is right. So minimum inhibitory concentration is depending upon the number of organisms present. Understand it? So if the number of organisms is present is more, right? Is the concentration is required the more. Understand it? If the number of organisms is very less, the concentration is required the very less. Understand it? So this is one of the disadvantages. Then time of exposure to the long, right? So how much time required for to inhibit the mechanism? This is the time of exposure. समजा जास्त असेल time of exposure तर it is a totally kill the mechanisms, and some of the disadvantages also is there. Then no sufficient organic matter is there. It is not containing any organic matter in this test. So this is a minimum inhibitory concentration. Then so this method is used for calculating the MIC. म्हणजे हे मिनिमम इनिबिटरी कॉन्सन्ट्रेशन आहे, वेगवेगळ्या डायल्युशन्स आहेत त्याच्यामध्ये, राईट? सो हे मिनिमम कॉन्सन्ट्रेशन आहे जेणेकरून ते ग्रोथ कुठलाही होत नाही. हे जे तुम्ही भेटलंय हे टेस्ट मध्ये थर्ड टेस्ट मध्ये राईट. कुठलंही जे मिनिमम कॉन्सन्ट्रेशन आहे त्या कॉन्सन्ट्रेशननी ग्रोथ होत नाहीये. We can inhibit all the mechanisms with this test. But after this, if you have reduced that concentration, is that, and after reducing the concentration, the growth of the mechanism is possible, right? So with this, what is the minimum? How much concentration is required for inhibit that organism should be know it. Then, so next test is used for that Chick-Martin test, right? So Chick-Martin's test is a modification of the radial Walker test is there. We have seen the radial Walker test, right? So this is the condition is which are mimicking naturally can using that the presence of organic matter. रेडियल वॉकर टेस्ट मध्ये कुठले ऑर्गॅनिक मॅटर यूज केलेलं नाहीये. पण ह्या चिक मार्टिन टेस्ट मध्ये तुम्ही ऑर्गॅनिक मॅटर यूज करू शकता, राईट. सो यू कॅन यूज द ऑर्गॅनिक मॅटर ॲज 3% dried human feces added into that test. Right? This is organic matter. So 3%. So the रेड लॉकर टेस्ट मध्ये कुठलाही आपण ऑर्गॅनिक मॅटर यूज केलेलं नाहीये, पण ह्या चिक मार्टिन टेस्ट मध्ये यूज करणार आहे. 3% ह्युमन पीसेस. Then contact time you can increasing. The contact time रेड लॉकरचा किती होता? Is a 2.5 minute to up to the 10 minutes. बरोबर. हा मिनिमम टाइम पिरियड होता. तर कॉन्टॅक्ट टाइम चिक मार्टिन टेस्ट मध्ये 30 मिनिटचा कॉन्टॅक्ट टाइम आहे. तर आपण ऑर्गॅनिझम कुठले यूज केले होते? Both is a similar TP is one of the organisms which are testing in the red Walker test. But in the Chick Martin test, we can use two organisms, the similar TP and Staphylococcus aureus. Right, these are the two things are used. Then, so what is the difference between the radial Walker test and Chick Martin test? So volume of medium is used for radial Walker test 5 ml. But in Chick Martin test, we can use as a 10 ml. Right. Then dilute of the test. What are the dilute is used in the radial Walker? Can use water. Is a normal water we can use, right? But in Chick Martin test, we can use as a yeast suspension. Understand it? So yeast suspension means contain the 3% of the organic matter can be used for that in the Chick Martin. Then reaction temperature is the 17.5 plus minus 0.5 degrees centigrade. So this is a reaction temperature required for radial Walker. Then Chick Martin test is only 30 degree should be required. Then organism is used Salmonella typhi in radial Walker, and Chick Martin is the Salmonella typhi as well as the Staphylococcus aureus you can use. Sampling time is 2.5, 5, 7.5 and 10 minutes. This last is 10 minutes. But in Chick Martin it is 30 minutes we can use. Then how you calculate that radial Walker coefficient? So how you calculate that coefficient? The dilution test is killing the 7.5. Means the that concentration within that 7.5 minutes, so all the organism is killed, right? Understand it? With that concentration, all other organism is killed within 7.5 minutes. So, but not killed now 5 minutes, right? It's the same for the phenol. So, it is for test, and this is for the phenol. Understand it? We have seen this formula again. Then the mean concentration of the phenol showing growth of after 30 minutes. So these are this comparing with that the phenol as well as your test, right? But time limit is 30 minutes. Is time limit but 30 minutes it can kill the organisms, but before that the 25 minutes, the 20 minutes will not be killed at all microorganisms. So this test is Chick Martin test, right? So then we'll say that this is about Kelsey and Sky's test, right? This is one more test. So capacity, we can use as capacity, use dilution test, right? So how much dilution is required for to make that solution, right? So this Kelsey and Sky's test, which are used as a triple challenging test. Why it is called as triple challenging test? We can use as a concentration, the fixed concentration of disinfectant. म्हणजे 10% concentration सगळीकडे असेल, right? But we can use organisms as a double. म्हणजे 3 टाइमला तुम्ही ऑर्गॅनिझम चेंज करू शकता, राईट? म्हणजे फर्स्ट टाइमला तुम्ही एक डायल्युशन कॉन्सन्ट्रेशन आहे 10% आहे, राईट? Then you can add the some of the 1% of the microorganisms into that. Then this is the first step. The second step, again, we can take that same concentration of disinfectant, then again added the 1% of that organisms. As such that we can repeat 3 times. right? Repeat 3 times. Then we can effective. So this disinfectant will be effective in a clean and dry condition. We can measure that two conditions are where there is clean condition and dry condition. Understand it? You can comparing the result with the clean and dry conditions. So effective in a clean and dry conditions, the dilution of the disinfectant is made with hard water. The test solutions, the dilutions are disinfectants तुम्ही hard water ने use करू शकता फॉर अ क्लीन कंडिशन. हे दोन कंडिशन आहेत. क्लीन कंडिशन एक आहे आणि दुसरी आहे तर डर्टी कंडिशन आहे. क्लीन कंडिशन मध्ये we can use a disinfectant solution with addition of the hard water. This is a clean condition, right? Then in dirty condition मध्ये we can use as a dilution of disinfectant can addition of the some of the yeast suspension for dry condition. Understand it? So these are the two conditions which are used for a clean condition and dirty condition. Then contact time of the disinfectant test organism is 8 minutes. All right. So one cycle is for 8 minutes. Then the test organisms we can allow, right? So maybe waste will be added, right? Waste will be added in the dirty conditions, and we can from the 0, 10, and 20 minutes interval we can contact with that. So depending on the disinfectant, we can use the single test organisms. Whether you can use the Staphylococcus aureus, there's a P. aeruginosa, the Pseudomonas aeruginosa, or Pseudomonas vulgaris, or we can use as E. coli. So any from this organisms we can use at a time. Understand it? Then, so this test is there. The test disinfectant for standard hard water. So disinfectant solution घ्यायचं, right? There varies concentration, 10% concentration म्हणा, and त्याच्यामध्ये तुम्ही hard water dilution टाकायचं. ठीक आहे? The hard water dilution टाकायचं. This is a test, right? Then in that test we can add the suspension of the test bacterium. त्याच्यामध्ये हे पहिलं की टेस्ट डिसइन्फेक्टंट झाला, त्यामध्ये हार्ड वॉटर डायल्यूट करायचं, म्हणजे तुम्ही कॉन्सन्ट्रेशन 10% करत असाल त्याच्यामध्ये तुम्ही टेस्ट ऑर्गॅनिझम ऍड करायचं आहे. बरोबर? इज सेम प्रोसिजर फॉर दिस इज अ प्रोसिजर फॉर द क्लीन कंडिशन, देन सेम प्रोसिजर वी कैन रिपीट फॉर that dirty condition. ठीक है? So after each addition of the organisms, the portion of the mixture can be removed, right? So after this we can after this addition of that microorganisms, right? And from that test we can take that some of this organism culture from that the basic test and again inoculate in the something, inoculate in the culture media. So culture media, soil culture media, we can use a test tube as well as there's a petri plate also you can use. So just we can make the ready for that test tube as well as that petri dish which are containing the solid agar media, right? हे ऑर्गॅनिझम तुम्ही घेतल्यानंतर याच्यामध्ये इनॉक्युलेट करायचं आहे सॉलिड मीडियामध्ये, आणि सॉलिड मीडिया इनॉक्युलेट केल्यानंतर वी कॅन इन्क्युबेट फॉर 37 डिग्री सेंटीग्रेड फॉर 24 आवर्स, राईट? याच्यामध्ये 24 आवर्स इन्क्युबेट केल्यानंतर यू कॅन ऑब्झर्व दॅट whether the any positive growth or not. म्हणजे ग्रोथ झालाय की नाही हे तुम्हाला फक्त बघायचं आहे. सो ग्रोथ झालाय, राईट? सो ग्रोथ व्हायला नाही पाहिजे मीन्स द टेस्ट इज पास. हे समजलं? कळेल तुम्हाला समजलंय? हॅलो हॅलो,
Yes, sir.
टेस्ट सोल्युशन आणि तुमचं टेस्ट सोल्युशन जे आहे ते इज अ क्लिअर सोल्युशन, द क्लिअर कंडिशन, आणि जे तुम्ही स्टँडर्ड सोल्युशन घेणार आहे इट इज अ डर्टी कंडिशन, राईट? तर हे दोन्ही सोल्युशन सेम मेथडने यूज करायचं आहे, राईट? तर डर्टी कंडिशन मध्ये हे आता क्लिअर कंडिशन आहे, बरोबर आहे? त्यामध्ये तुम्ही सस्पेन्शन ऍड केलंय बॅक्टेरियांचं, आणि त्याच्यामधून इनॉक्युलेट करून तुम्ही कल्चर मीडिया तुम्ही इनॉक्युलेट केलंय आणि तुम्ही इन्क्युबेट केलंय इन्क्युबेटर मध्ये 37 डिग्री सेंटीग्रेडला. Okay, and then after we can observe that growth. Okay, a same procedure we can use for the dirty condition. Okay, so this is about the skies and tests which are used for that. So disinfectant is evaluated for its ability to kill the organism. It means so how much concentration. So disinfectant means the concentration is fixed, but we can use that is a different concentration of the organisms. The triple time concentration we can use for the organisms, but the disinfectant solution concentration is fixed, but there is a organism concentration is different. Understand it? So whether there's growth of organisms or not, so this is the test is called as a pass or fail. It's not a coefficient. Is one coefficient, two coefficient not there. So whether the test is pass or not, right? So this is about the Kelsey-Scar test, right? So we'll see one figure, one video, then you understand better. Hello? This is the Hello?
Yes, sir.
Yes, sir.
So we have already seen that radial occur test. So radial occur test is have limitations. So no organic matter we can use for that. ठीक आहे याच्यामध्ये कुठले ऑर्गॅनिक मॅटर रेडियल वॉकर मध्ये यूज करत नाहीये एकच ऑर्गॅनिझम तुम्ही यूज करू शकताय राईट याच्यामध्ये आणि टाइम ऑफ पिरियड इज अ व्हेरी शॉर्ट फक्त 10 मिनिटाच्या वर हे टाइम पिरियड नाही सो दिस इज अ लिमिटेशन ऑफ द रेडियल वॉकर टेस्ट देन Then Chick-Martin test is there. Chick-Martin is performed with a similar way of the radial occur test. So some of the little variations is there. So in that variations is there, 3% of organic matter can be used. We have seen that. So the dried human feces we can use for that as a organic matter. Then, so Chick-Martin coefficient will be calculated by the dividing the concentration of the test disinfectant by concentration of the phenol. The similar absence of the growth. राईट म्हणजे 5% कॉन्सन्ट्रेशनला कुठलाही ग्रोथ नाहीये असं तुम्ही दोन्ही टेस्ट आणि फेनॉलचं कम्पेअर करून त्याचं चिक मार्टिन कोएफिशिएंट तुम्ही काढू शकता ठीक आहे सो दिस इज अ सॉरी ठीक आहे सो दिस चिक मार्टिन कोएफिशिएंट इज sorry ठीक है so the Chick-Martin coefficient is concentration of test disinfectant that kill in a minimum concentration ठीक है is divided by concentration of the phenol that kill the microorganisms with a minimum concentration ठीक आहे हे दोन कॉन्सन्ट्रेशन तुम्ही घ्यायचं आणि त्याचं कोएफिशिएंट तुम्ही डिटरमाइन करायचं तर ऍक्च्युअल प्रोसिजर बघूया आपण ठीक आहे okay so this is a test solution so the 3% organic matter we can use we can add the 100 ml of water into that test solution okay and they prepare the test tube that each test tube is containing the 2.5 ml okay 2.5 ml of the there is a disinfectant solution so take 2.5 this is a 2.5 ठीक आहे 2.5 मध्ये काय 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 आहे त्यामध्ये it is containing the organic matter 2 ml of the microorganisms and 3% of organic matter okay the 3% organic matter हे सगळं तुम्ही याच्यामध्ये टेस्ट मध्ये ऍड केलाय ठीक आहे ऍड केल्यानंतर you can use ठीक आहे ते मार्कोऑर्गॅनिझम जे यूज करायचे तुम्हाला 24 आवर्स ओल्ड कल्चर पाहिजे राईट आणि ते कल्चर तुम्ही त्याच्यामध्ये तुम्ही ऍडिशन करू शकता 2 ml इच टेस्ट ठीक आहे त्यामध्ये 3% ऑर्गॅनिक मॅटर तुम्ही ऍड करू शकता प्रत्येक याच्यामध्ये त्याच्यानंतर ठीक आहे. So these are different dilutions of phenol. ठीक आहे. तुम्ही डायग्राम बघू शकता, डिफरंट डायल्युशन ऑफ टेस्ट सोल्युशन. ते फिनॉलचं इथं बनवलंय आणि टेस्ट इथं बनवलंय. प्रत्येकामध्ये 2.5 ml तुम्ही टेस्ट तुम्ही ऍड केलाय. ठीक आहे. त्याच्यामध्ये अगेन 2.5 ml मॅक्रोनिझम तुम्ही ऍड करायचंय प्रत्येक टेस्ट मध्ये आणि 3% ऑर्गॅनिक मॅटर मिक्सर ऍड करायचंय. हे मिक्सर ऍड केल्यानंतर अलाव कॉन्टॅक्ट टाइम फॉर 30 minutes. हे सगळं ऍड केल्यानंतर आय कॅन अलाव कॉन्टॅक्ट टाइम फॉर 30 minutes in between. Okay, then the phenol organism, then we can transfer that each and every test tube from that into that mixture of the 10 ml. This is a recovery culture media. Okay, recovery culture media and culture media मध्ये तुम्ही ट्रान्सफर करायचं तुम्ही इनॉक्युलिंग लूपनी प्रत्येक टेस्ट याच्यामधून आणि you can incubate for 37 degrees centigrade for 48 hours. तुम्हाला राईट फेनॉलचं पण तसंच आहे आणि डिफरंट सोल्युशनचं पण टेस्ट डिसइन्फेक्टंट सेम सोल्युशन आहे. ठीक आहे. समजला या. ठीक आहे. Then observe the concentration and the taste disinfectant we can kill organisms. तर कुठलं कॉन्सन्ट्रेशन ने किल होतात आणि कुठलं कॉन्सन्ट्रेशन किल होत नाही हे तुम्ही डिटरमाइन करायचं आहे. ठीक आहे. सो दिस अबाउट the Chick-Martin test. Then next test is the Kelsey and Scar test. Kelsey and test it is a triple challenge test is there, and test is we can check is 3 times with the successive addition of the microbial suspension. म्हणजे टेस्ट डिसइन्फेक्टंट आहे एका वेळेला ऍड करायचंय आणि त्यामध्ये तुम्ही 3 times microbial suspension तुम्ही ऍड करायचं. ठीक आहे. सो टेस्ट is performed in both this condition, clean condition, ऑर्गॅनिक मॅटर, नो ऑर्गॅनिक मॅटर, डर्टी कंडिशन मध्ये ऑर्गॅनिक मॅटर ऍड करायचं. 3% ऑफ ऑर्गॅनिक मॅटर वी कॅन ऍड इज इन डर्टी कंडिशन. सो दिस 2 conditions वी कॅन यूज फॉर दॅट. ठीक आहे. नंतर सो ड्युरेशन ऑफ टेस्ट इज 30 minutes. Duration of test is 30 minutes. The one organism is used for the study of these are the organisms, P. aeruginosa, Vaginalis, Staphylococcus aureus, and E. coli. These are the 3 organisms. So each from that the one organism we can use. So interpretation of result is results are reported as a pass or the fail. म्हणजे result is not containing any coefficient. म्हणजे हे ड्राय कंडिशन मध्ये क्लीन कंडिशन मध्ये can use water with the test organism. Right, and incubate for 48 hours. Dry condition मध्ये organic matter use करायचं, right, with this your concentration, this your organic matter. So both these conditions, whether how much concentration we can growth of the organisms or not, right, and just interpret the result with the pass or fail. So this test is the pass or fail. Just we can observe that. Okay, you see in the diagrammatically. Okay, so this is a disinfectant. This disinfectant is 3 ml. And the microbial cell is 1 ml. Okay, त्याच्यामध्ये तुम्ही ऑर्गॅनिक मॅटर यूज केलेला नाही. So after 10 minutes, 8 minutes, total time is 8 minutes. Right, हे फर्स्ट राउंड आहे. त्याच्यामध्ये टेस्ट डिसइन्फेक्टंट ऍड केले 3 ml आणि मॅक्रोबियल सेल ऍड केले परत 1 ml. ते प्रत्येक याच्यामधून ट्रान्सफर केलंय प्रत्येक ग्लास स्लाइडला, प्रत्येक याला पेटी डिश मध्ये. पेटी डिश इज ऑलरेडी कंटेन दॅट द agar medium which are solidified. ठीक आहे. हे ट्रान्सफर केल्यानंतर you can check that whether the growth or not. Then so this is the first round. The second round is for after the 18 minutes. Okay, after so this is for 8 minutes, and this second for the after the 18 minutes, sorry. So then again the test disinfectant तुम्ही ऍड केलाय पहिल्यांदाच, right, ते स्टॉक सोल्युशन इथं घ्यायचं आहे आणि त्यामध्ये अगेन we can add the मॅक्रेबल सोल्युशन 1 ml. म्हणजे टेस्ट डिसइन्फेक्टंट इज फिक्स आहे, फक्त मॅक्रेबल सेल वी कॅन ऍडिशन ऑफ 3 times. ठीक आहे. So again the transfer of the microorganism, again there is same procedure for that. So each and you take that microbial organisms, take that test organisms and just inoculate into that the agar medium. Okay, so this is for 18 minutes. Then after this for 20 minutes. So 28 minutes, the 3 times we can use for that. The 28 minutes, the same procedure we can use for that. Okay. Again, we can take that same disinfectant solution as a 3 ml, and again we can add that the microbial cell 1 ml. Okay, so the 3 times to me microbial cell add killer, and after the 20 minutes, then we can take that inoculate from that and just transfer into that the agar medium as the various agar veggie dish. Okay, then after that this is for the first step is for 8 minutes. Then second step for the 18 minutes, and the third step is for the 28 minutes. Okay, so this is about the skies and tests are there. So we can determine that. So how much concentration, right? How much of the concentration of disinfectant to kill the microorganisms, and that concentration we can see that whether this test is fail or not. Okay. Okay, I understand this. Hello.
Yes.
Yes, sir.
Hello. This is the first slide here. Antimicrobials.
Yes.
Yes, sir.
Yes,
So
sir.
we'll see some of the small point of that antimicrobial property of the disinfectant. What are the bactericidal and what is the bacteriostatic, right? So we can determine whether this substance is bacteriostatic or bactericidal or not. We can determine it. We can use the different methods for that to determine this. So evaluation of the bactericidal and bacteriostatic activity. ठीक आहे. समजा की तुम्हाला एखादा सबस्टन्स आहे, अँटीबॅक्टेरियल सबस्टन्स आहे, पण त्याची ऍक्टिव्हिटी माहित नाहीये. ठीक आहे. कुठल्या कॉन्सन्ट्रेशन ऍक्टिव्हिटी दाखवत आहे हे तुम्ही डिटरमाइन करणार आहे. The bacteriostatic is to inhibit the growth of the microorganisms, but the bactericidal is to killing the microorganisms. Okay, we have already seen that. So general principle of antibacterial laboratory is to inhibition of the microbial growth under the standard condition which are used for that, right? So means that with that condition, with that concentration of the antibacterial agents, so can kill the microorganisms and see that these are the concentration which give that therapeutic efficacy of that particular antibiotic. म्हणजे अँटीबायोटिकचा डोस आहे 100 मिलीग्राम आहे डोस आहे. तर 100 मिलीग्राम डोस मध्ये टोटल मायग्रेझम शुड बी किल. बरोबर. सो तो डोस तुम्ही कॅल्क्युलेट करायचा आहे. समजा की ते 100 मिलीग्रामला किल होतात मायग्रेझम, तर 400 मिलीग्राम त्याला द्यायची जरूर नाहीये. सो कॅन डिटरमाइन दॅट हाऊ मच concentration of that particular antibacterial agents, right? So we can kill the microorganisms totally. Understand it? So any change in that the antibiotic molecule, right, will not be detected by chemical method, right? समजा एखादा अँटीबायोटिक मोलिक्युल आहे, काहीतरी मॉडिफाय केलंय, त्याला चेंज केलंय, पण त्याचे तुम्ही जे चेंज केलेलं आहे त्याची ऍक्टिव्हिटी काय आहे तुम्ही केमिकल मेथडने करू शकत नाही. तर यू कॅन यूज एज अँटीमायक्रोबियल ऍक्टिव्हिटी. म्हणजे अँटीमायक्रोबियल ऍक्टिव्हिटी तुम्हाला कळेल की किती कॉन्सन्ट्रेशनचा डोस आहे, अँटीबायोटिक डोस आहे, त्यामुळे किती प्रमाणात ते antibacterial mechanisms kill hota hai. Barabar? So this is called as, so the microbial assays can be used for to resolving the doubt regarding that. So how much concentration used as the antibiotic to kill the microorganisms. Understand it? So that should be know that antibacterial activity. Then, so these are antibacterial agents which are used for to prevent the bacterial infection by the killing or preventing the growth of organisms. Right? Prevent the organism, preventing growth of bacteria as well as killing the that growth of the microorganism. So these are the different terms we can use. Bactericidal. Bactericidal is chemical agent which are capable to killing the bacteria, right? But not necessarily killing the spores. So bactericidal agents are not be kill the spores, but they can kill the bacteria, right? So bacteriostatic is we can chemical agent which are capable to preventing the growth of microorganisms, but not killing the microorganisms. Understand it? So this is called as the bacteriostatic. So minimum availability concentration we already said. So lowest concentration of the drugs or any antibiotic should be inhibit the growth of the microorganisms. With the lowest, we should be know that. So how much lowest concentration will be inhibit the growth of the microorganisms or not? This is called as a minimum availability concentration. Then minimum bactericidal concentration. So minimum concentration of a drug or antibiotic can kill that is a given test organisms. Right. So कुठल्या प्रकारचे organisms you can kill करतात ते antibiotic. So you should know that because all type of organisms are there. Maybe positives are there, negatives are there, right? So some of the antibiotics we can kill only the negative organisms. Some of the antibiotics we can kill only the positive organisms, right? So you should know that. So with that test, with that antibacterial agent, so which type of the organisms you can kill, right? So this is called as minimum bacterial concentration. Then this is our in vitro and bacterial activity screening. This antibacterial activity will be this bacteriostatic and bactericidal. All right. So bacteriostatic, we have already seen that the different method we can use: serial dilution, fluid media, serial dilution in the solid media, the cup plate method, gradient plate method, and dish plate method. All right, understand it. So generally, this is this test can be conducted and to see that bacteriostatic activity of the particular antibiotic or antibacterial agents. Right? Whether that whether that bacteriostatic agents are which is preventing the growth of the microorganisms or not will be determined with the help of these methods. Right? Then the bactericidal activity, whether this antibacterial agent we can killing the microorganisms or not, can determine with these two methods can be used. So this is a method 1 and method 2. Right? So method 1, we already said that the phenol coefficient test. So the phenol coefficient should be 1. So is give that same effect of the test disinfectant is comparing with the phenol disinfectant. Right? Is more than 1. So means that disinfectant solution is higher activity than the phenol. Understand it? So you can calculate with the extension time, means how much time required for kill the organisms and how much concentration are required for the kill the organisms. These are the two parameters will be calculated with this bactericidal activity. Right? So we'll see in brief about this. Right. So this is a agent we have already seen that serial dilution, fluid media methods or the solid media, cup plates, the gradient plates and dish plates. We have seen a brief about this. Serial dilution, we have already seen that. In that, we can calculate that a minimum availability concentration, right, with the taken the bactericidal activity, right. So this is a stock solution. Can prepare the stock solution is contain the various disinfectant solution, right. From the disinfectant solution, we can add the 1 ml from this. From that 1 ml, we can add again the second, then again the 1, again the third, and again the fourth, right. So you can concentration of that disinfectant solution is right is a 1:10, 1:100, 1:1000. हे स्टॉक सोल्युशन आहे. स्टॉक सोल्युशन मध्ये 1 ml तुम्ही ऍड करायचंय, त्याचं कॉन्सन्ट्रेशन चेंज होईल, राईट. तर कुठल्या कॉन्सन्ट्रेशनने इनहिबिट द ग्रोथ ऑफ मॅग्नेशियम होईल, यू कॅन डिटरमाइन इट, right. So the same method you can dilute with that, and then after dilution can take that test solution and just inoculate on that solidified surface of the agar medium. Find it. The same method we can use for that. So prepare the that is a serial dilution. Means we can prepare the bore in that bore, right, and keep for incubate for 2 to 3 days. And after incubation, see the growth of the microorganisms or not, or whether this turbidity or non-turbidity, whether the solution is clear or not, or whether the turbid or not. The turbid means there is growth of the microorganisms, and non-turbid means there is a no growth of microorganisms. This is covered as the couplet method. We already said that. Can prepare that. Take that, so that is agar medium, molten agar medium. Just pour into that the petri plate, right? And after solidification of petri plate, you can prepare the bore. So bore with the help of this instrument and prepare the bore into that. And after preparation of the bore, right, then you can addition of there is a disinfect solution into that bore, and test organisms you can just spread on that surrounding surface of that the agar medium and incubate for 24 hours or 32 minutes. 24 hours, and after that incubation, see the growth. This is the growth of the organisms, right? So the growth of organisms means that the test solution, the test solutions are is a less abilities to kill the organisms. ज्यामध्ये the growth nahi hai means that concentration we can kill the organisms, right? And that concentration is we can use as an antibacterial concentration. So this is about the couplet method. Then this gradient plate method, we have already said that it is the first is can take that petri dish again. Then first they can molten mass at the nutrient agar and just keep that the plate at the inverted position, right, or graded as elevation position, so can prepare that the first base. Then again add agar medium with some of the your antibiotic solution. Okay, हे first layer solidify झाल्यानंतर can add the second layer is contain the agar medium as well as the antibiotic solution or any disinfectant solution. त्याच्यानंतर you can take that the test organisms can spread on the surface of that agar medium, and after that you can see that whether the growth of the organisms or not. That's right. It. So this then there is a ditch plate method. The same you can we use for that. Prepare the ditch with the help of this knife, right? With the cut it that solid surface of the agar medium and prepare the ditch. In that ditch, we can add that disinfectant solution or antibiotic solution, and around that you can spread that microorganisms with the help of the inoculating glove and incubate for again 24 hours or 42 hours for 37 degrees and incubation, and see the whether this zone of emission or not, right? So if there is zone of emission is not there, right, so means the test organism is it's having abilities to kill the organisms, right? Growth ही जास्त असेल तर म्हणायचं की test organism is not appropriate to killing the microorganisms. That's not it. So this is called as the ditch plate method. We already said that. Then the bactericidal agents. So this all the tests we can use for the bacteriostatic agents, so preventing the growth of microorganisms. Then this method bactericidal agents we can kill the microorganisms, right, within a short period, but including the spores also. All right. So assessment of the bactericidal activity, right? So this is a assessment of the bactericidal activity. This the end point is extension time. So how much time required for the killed organisms we can determine with these two methods can be used. Is the first is a phenol coefficient test. We already said that. So how much extension time is fixed? The time is fixed. We have seen that the 2.5 minutes, 5 minutes, 7.5 minutes, and the 10 minutes. And from that 7.5 minutes we can kill the organisms, but 5 minutes you cannot be killed the organisms. You can calculate that the phenol coefficient of that phenol. Can comparing with the phenol. Then the method 2 is used as the concentration of bactericidal which are fixed. The extension time can be different. Right. So we have seen that Skygiant test is there. In that Skygiant test, Skygiant scale test, right? So in that test, right? So we can concentration of the bactericidal is fixed, but we can add that the 3 times the concentration of the test organisms and determine that. So how much time required for killed organisms? For the first step is that is 8 minutes. The second step is 18 minutes, and the third step is at 28 minutes. From that, we can determine that So how much concentration is we can kill the organisms or not. So this is about the bactericidal activity. We have already seen that. Right? So that we have already seen. So Chick-Martin test, the highest final concentration inhibiting and the lowest permitting dose. म्हणजे कुठल्या कॉन्सन्ट्रेशनने म्हणजे किल होतात ऑर्गॅनिझम्स आणि कुठल्या कॉन्सन्ट्रेशनने किल होत नाहीत. We can determine with phenol and the test divided by test. Right. So these are the different tests we can determine, and to see that see that is a we can determine see that is a whether that antibacterial agent having that bactericidal activity as well as the bacteriostatic activity. The bacteriostatic agents we can use for different tests we can see, and the bactericidal we can see that Riedel-Walker test or Chick-Martin test and the SCART test is used for that whether this antibacterial activity or not. Right. So this is about the test we can conduct. Then we'll see one more thing. Hello, this is slide. Hello?
Yes, sir.
So we'll see
Yes,
the
sir.
one point is that what is the mode of action of the mechanism? So how the bacteria we can kill. Understand? We already said that the point, but we can just refreshing of that the point. So mode of action means how much concentration is required for the killing the organisms and which type of the organisms which are killed. So what are the mechanisms we can kill the microorganisms are there? So general method we can see that the we can use a different disinfecting solution. So just we can denaturalize on the proteins in the cell wall or the cell membrane. And after denaturalization of the proteins in that cell membrane, so we can distort that the cell membrane or bacteria. So these are the different action of the cell. So if I use a different antibiotic solution or disinfectant, it be acting on the external membrane of the bacterial wall. Right? So wall is composed of the phospholipids and the lipopolysaccharides. It will be stabilized with magnesium and cations, calcium cations. Right? His membrane कशामध्ये बनवलं आहे? The phospholipids and lipopolysaccharide पासून, right? And it can stabilize with the magnesium and the calcium cation. What if I use as a disinfecting solution? Disinfecting solution, it can acting on the bacterial cell wall, right? And after acting on the bacterial cell wall, it will be absorbed on that bacterial cell wall, right? Absorb on the bacterial cell wall. After absorption, can destroy the cell of that the cell membrane. Destroy that cell membrane, right? Because it will disturb that phospholipids and lipopolysaccharides, and what are the stabilized magnesium cations are totally disturbed. After the ionization of the disinfecting solution, so the solution will be absorbed and that the cell membrane as well as repel the cell membrane and it will be destroyed at your cell membrane. And this is one of the mechanism. Then action of the bacterial wall. So the bacterial wall is the rigidity and the differ which consider with the gram-positive and gram-negative bacteria, because the gram-positive bacteria its property is different, gram-negative bacteria's property is different. So rigidity and the differences in between that is there. So the different concentration, different agents, antibacterial agents must be required for killing the microorganisms. So we can determine that the bacterial wall also. Then action of the cytoplasmic membrane. Some of these antibacterial agents we can directly attacking on cytoplasmic membrane, right? So this cytoplasmic membrane, we can penetrate that that disinfecting solution to that cytoplasmic membrane and can destroy that cytoplasmic membrane. Now, after destroy that, right? So there is no survival of that bacteria. So this is about then action on the energy metabolism. Sometimes some of the disinfectant will be mechanism is to disinfectant will be acting on adenosine triphosphate production. So it's a totally synthesis of ATP is stopped. And as well as that ATP which are used as energy for all the living organisms. If the ATP is not there, so the ATP is energy, all right? So if we are stopping that synthesis of ATP production, right? So ultimately there is a no survival is possible for that. Then action on the cytoplasm and the nucleus, separate cytoplasm and nucleus at the chromosomal level. So if you're acting on the cytoplasm and nucleus, it will be again it changes in the chromosomal level, and after changes in chromosomal level is a total activity, total that is originality of that macroscopic cells is not there. Then the action of the bacterial spores. Some of the antibacterial agent will be acting on the spores only. So can kill the spores, right? Because the normal antibacterial agents is not be kill the spores, but special antibacterial is required for kill the organisms are there. So why the spores are not be killed? Because it's the presence of the dipiclonic acid in bacterial spore. Understand it? Doses dipiclonic acid असल्यामुळे राईट कुठले डिसइन्फेक्ट सोल्युशन तुम्ही सेल वॉल मध्ये पेंटेड होत नाहीये. पेंटेड न झाल्यामुळे will not be, it is a no question of the distort that the spores. Understand it? So with this dipoclonic acid is a bacterial, this is a dipoclonic acid in the bacterial spore is one of the important aspects to make the cell is impermeable. So means any antibacterial agents are not be penetrated to the bacterial cell, and there is no question of destroying that bacterial cell, right? So it's make them more resistant to that disinfectant solution. But if I use as some of the oxidizing agents, there is a hydrogen peroxide as well as a chlorine, right? We can oxidation of that dipoclonic acid. After oxidation of dipoclonic acid, then can change the originality of the property of the dipoclonic acid and the permeability of the disinfectant solution in that bacterial cell and can kill that spores again. Understand it? So these are the different mechanisms you can. This is a use for that to kill the microorganisms are there. Do you understand this? Hello? Hello? Hello?
Yes, sir.
Listen, ours.
Yes, sir.
Okay, we'll stop here today. Okay, we can leave.